The next speaker is Dr. Edward Breitschwerdt. He is a professor of medicine and infectious diseases at North Carolina State University and an adjunct professor of medicine at Duke University Medical Center. He directs the Intracellular Pathogens Research Laboratory in the Center for Comparative Medicine and Translational Research at North Carolina State University and co-directs the Vector-Borne Diseases Diagnostic Laboratory. His clinical interests include infectious diseases, immunology, and immunology. His research has emphasized vector-transmitted intracellular pathogens. He has contributed to cutting-edge research in the area of animal and human
Bartonellosis.
Thank you.
You're welcome.
Ladies and gentlemen, will you please welcome Dr. Edward Breitschwerdt. Thank you.
Good morning, everyone, and thank you to NorVect, the organizers, for being here, and most importantly, for the very kind and complete communications that managed to get me across the ocean to this meeting. The hospitality has been absolutely wonderful. Over the years, my laboratory, starting in the early 1980s, has worked on anaplasmas, ehrlichias, babesias, bartonellas, quite a few of the vector-transmitted organisms in predominantly dogs, to a certain extent some other animals. And then we transitioned, for reasons that'll become obvious, into studying human Bartonellosis. So the disclosures until recently, I had no conflicts of interest in my life other than the fact I couldn't get research funding. More recently, I have a conflict of interest, which is a company that we started called Galaxy Diagnostics. All the publications that will be listed here are publications that emanated from the College of Veterinary Medicine at North Carolina State University from the research group that's there, and hopefully nothing that I'll say will reflect a conflict of interest relative to the company. It's very interesting, as I sit and I listen to the other speakers, in regard to similarities between Bartonellosis and borreliosis, and I think one of the challenges we're going to all face is separating out what is Bartonellosis, what is borreliosis, and how many of the patients out there actually have both of these infections. And I think I'll be able to convince you that we have some sorting out to do in just a little bit. The other aspect that makes these alpha proteobacteria very unique is their ability to invade many cells within the body. And as we'll see in a cartoon in just a minute from Christophe Dieho from Switzerland, these bacteria initially penetrate the skin, interdendritic cells, but once they get into the body, they actually localize to the endothelial cells, then invade in cycles, as Joe talked about in regard to Borrelia, the erythrocytes, and they can circulate within the body in erythrocytes, monocytes, and at least when Kempf published the study a few years ago, these are the only bacteria that were known to infect CD34 progenitor cells within the bone marrow, which I think may have some clinical importance in the context of bone marrow disease. So I put this slide together about a year ago because I think it reflects the state of Bartonellosis. And I had to take some solace from the comment from Aristotle that it's the mark of an educated mind to be able to entertain a thought without accepting it. Because as a veterinary researcher looking at Bartonella, my concern is, could there be a hidden day modern epidemic? And this is perhaps very pertinent to those of you in Europe realizing that when the bubonic plague occurred in the 1400s, which is credited with killing about a third of the European population. We didn't know that it was a bacterial infection. We didn't know that it was being transmitted by a vector, and we didn't know that there was an animal reservoir in the context of rats that were responsible for this. People actually thought that it was divine intervention that had brought this plague along. And so as I've thought about Bartonellosis, my initial question was, well, how in the world can you hide an epidemic from epidemiologists, microbiologists, and clinicians? And honestly, when I first made this slide, I said, perhaps it's very hard to do that, and you'll see I changed that to perhaps it's not so hard. Essentially, you start with an unknown genus of bacteria, and if it were not for the AIDS epidemic, I would not be giving this lecture today, as we'll see in a minute. Secondly, as Stanley Falco, the past president of the American Society of Microbiology, stated, Bartonella is a stealth bacteria. It is capable of flying under the radar and is very difficult to detect in patients, as you'll see in just a little bit. From the standpoint of a veterinarian, I'm very concerned because every day we're finding that the wildlife reservoir for these bacteria is absolutely tremendous. Essentially, squirrels, kangaroos, and many, many other animals, cats, dogs, deer, all are running around in the world with their own Bartonella species that are vector transmitted and maintained in an enzootic cycle, but also some of these, as you'll see in a minute, make the jump out of that animal to infect a dog or a human and cause disease. And then what's becoming more disconcerting is the fact that we're finding an increasing number of vectors that are competent for transmitting these bacteria, and we'll look into that as well. So as I alluded to, what happened in the mid to late 1980s is that physicians saw bacillary angiomatosis and pileosis hepatis in AIDS patients. They then, using silver stains that Alan mentioned yesterday, were actually able to see these bacteria and large numbers of bacteria in the tissues of these lesions, but microbiologists were not able to grow them. David Relman, a physician at Stanford, used PCR targeting the 16S ribosome RNA gene, and all of a sudden, these bacteria, based on DNA sequence, became either Bartonella quintana, the trench fever agent, which you're very familiar with, or a new bacteria, Bartonella henselae, which becomes the cat scratch disease agent. Now, I put this slide in next because it took us 10 years to then identify bacillary angiomatosis in a dog that a veterinary internist was immunosuppressing because of bone marrow disease. This dog had pancytopenia. It was thought to be an antibody-mediated process, so the treatment was immunosuppression, and I'll come back to this again in the lecture in the context of endocarditis. This dog went on to develop cutaneous lesions. The dermatopathologist that reviewed the biopsy, from now a veterinary dermatologist, said this looks like human bacillary angiomatosis, and we were able to grow Bartonella vinsonii berkhoffii, which is a bacteria I'll introduce to you here in just a minute, from this and demonstrate the organism in the tissues. So the other question I asked as a veterinary internist, we see a tumor called an hemangioparasitoma that has a very unusual biological behavior in dogs. And one of the graduate students in my lab, we started culturing dogs with hemangioparasitomas to make an isolate of this bacteria. Subsequently, as you can see in the one reference in the Journal of Clinical Microbiology in 2009, a physician contacted me because his son had an epithelioid hemangioendothelioma. It took me about a year to be able to say that without stumbling across it. And he was reading the literature, and he found that patients with this tumor have high levels of vascular endothelial growth factor. Bartonella has a very unique ability to induce the production of vascular endothelial growth factor. So when he contacted me, he asked me, did I think it was possible that these two things could go together? In blood culturing this boy, we were able to isolate Bartonella vinsonii berkhoffii from his son, as well as demonstrated in the tissues. And we've worked very hard and are getting a little better at demonstrating this organism in the tissues. So the role that Bartonella plays in regard to vascular disease and vasoproliferative tumors, I think we're just starting to ask the question, how important is this? But certainly there's some references saying there may be a role here, and we've now identified EHE patients from England, Australia, and the United States that are infected with a Bartonella species. So the next important thing after AIDS was Russ Rigneri, a rickettsiologist at the Centers for Disease Control, made the association between cat scratch disease and this new bacteria that Relman had PCR amplified out of AIDS patients that becomes Bartonella henselae. And again, for all the physicians in the audience, you're familiar with cat scratch disease. I actually, for many reasons, do not even like that term and would hope that people would start to use Bartonellosis rather than cat scratch disease, and hopefully the reasons will become obvious as we go forward. But an inoculation papule, a large lymph node, exposure to a cat and a fever is the classical cat scratch disease. And one of the problems I have with this is everyone considers this a self-limiting disease. but work from physicians in Israel, as well as work from our laboratory, suggests that this is not uniformly true in all patients. It's likely that most people clear this infection, but all patients do not. So the next important thing that happened, so we did AIDS, we did cat scratch disease very, very quickly, and was Bartonella endocarditis. And you can see on this slide that starting in 1993, 3 cases in the human literature identified different Bartonellas in association with human endocarditis. What you can also see in this slide, which I'll emphasize on the subsequent slides, is that the dog is developing endocarditis that historically we would have all put into that category of culture-negative endocarditis, similar to Coxiella burnetii, and that the dog is developing endocarditis with the same Bartonella species that we tend to find in association with human endocarditis, okay? And I think the dog is a great naturally occurring model for the human disease, and I actually think the human is a great naturally occurring model for me to know what to look for in dogs. So, no offense to any humans that might be in the audience. So, one of the issues that then evolved between 1993 and 2013 was, okay, Breitschwerdt, and these are my colleagues as veterinary internists, would say, you're really excited about this new genus of bacteria, you keep publishing these little case reports or case series. How do we actually know this thing is causing anything in patients? And as I thought about this, one of the things that I have an advantage as a veterinarian is I can look at the way an infectious agent behaves across species. And so this postulate, which is like any other postulate, it doesn't prove anything, but it helps us try to give strength to associations, suggests that if you can find the same pathology, and you can actually find a pathologist or 3 pathologists that agree that that's the same pathology, in a cat, dog, horse, and human, and you find the same bug, well, maybe there actually is an important disease association there. And for endocarditis, granulomatous inflammatory disease, chronic intravascular infection, and to a lesser extent, vasoproliferative tumors, we can make these arguments, and some of the reference that I've showed you will do this. So from the top of this slide with Pileosis hepatis and bacillary angiomatosis on down through encephalitis, I think the evidence is fairly strong that there is association between Bartonella and these disease entities. We see a tremendous amount of immune-mediated thrombocytopenia and immune-mediated hemolytic anemia in dogs. And the reasons for this are often Babesia infections or Ehrlichia infections, and we believe Bartonella infections, although the study that needs to be done to prove that has not yet been done. But I think that one's pretty likely as well. So the point I would make is that from my standpoint as a veterinary internist that's responsible for kind of cats, dogs, horses, what have you, the dog seems to be not only our best friend, but maybe the one that immunologically tends to develop the same type cancers, the same type diabetes, and perhaps some of the same types of changes when experiencing infectious disease. Now, one of the other problems I alluded to earlier in the context of the genus Bartonella is we went from Bartonella being transmitted by fleas and by the human body louse to Bartonella being transmitted by KEDs or wingless flies, by biting flies, by the human body louse, and by other vectors that we'll talk about here in just a minute. The primary mechanism by which all these Bartonellas, this is from Deho's cartoon, is the sandfly or the flea, the sandfly through a bite, the tick most likely through a bite, and the flea and the louse by depositing infected feces. So they did studies 100 years ago when trench fever was studied, showing that if you applied louse feces to a scarified human skin, those volunteers would go on to develop classical trench fever. Again, that was a time when we didn't even know it was a bacterial infection. Everyone assumed it was a viral infection. What is true in the rodent model is that it's probably dendritic cells that carry these organisms to the endothelial cell. And then what the Bartonella researchers are looking for is what's the primary niche in which these bacteria are hiding. Some believe, as the AHO, that it's the endothelium. The French researchers believe it's the bone marrow. and I believe it's probably multiple tissues in the body in which these bacteria are capable of hiding, and it's not a single niche. But the problem is if you do this in a rodent model, you'll get a very predictable bacteremia. Trench fever was called 5-day fever, and you'll get about a cycle that's every 7 days in a rodent model. When we do this by intravenous blood transfusion in cats, we have had cats that were bacteremic for 3 weeks, were non-bacteremic for 90 days, and were bacteremic again. So that makes a very unpredictable pattern of bacteremia, at least in some cats with some Bartonella strains, and certainly is an area in which we could use a little more work. Now, the other area that has influenced my life is telephone calls and emails from sick people in the middle of the night. And this is an example where we were asked to do some testing of a family in Kentucky because 3 of the individuals in the family had developed neurologic disease. And again, I believe neurobartonellosis is probably the most important overall manifestation of this particular genus, both in animal and human patients. And what we ended up finding in this story was this family lived in an apartment. That apartment had become flooded. When that flood occurred, these, what we call in the United States, these little roly-polies or wood lice grew in the apartment. There's a spider that is called a woodlouse hunter spider that then came into the apartment, and the mother, fortunately, 18 months before we became involved, took photographs of the spider bites on her children. The youngest boy developed Guillain-Barré syndrome, then went on to the diagnosis of chronic demyelinating polyneuropathy, and we grew Bartonella henselae out of the mother and the 2 sons. So we also, the father, who was obviously very vested in what we were doing at that point, went back and collected these 2 guys 18 months after this all occurred and they had moved out of the apartment. And when I say we, this means really the researchers in the lab, not me, that are doing this work, were able to amplify the same Bartonella henselae genotype out of both woodlouse and the spiders. So this is a little unprecedented and obviously was a little hard to get published, but there is the possibility there's other vectors out there that are confusing our life. Now this is a painting by a Spanish artist that really depicts my career as a veterinarian. I'm responsible, as are my colleagues, for all the medical care of all the species on the planet except for one, that's you guys. And we actually, in our oath, have an obligation to help protect human health also. And unfortunately, Bartonella has completely ruined this picture for me in the context that if you dump Bartonella and rodent in, you will find that we're probably up to 20 species of Bartonella that now have official names in rodents. We went from not knowing these bacteria existed in North America in any animal or any human essentially in 1990 to now over 30-some named species of Bartonella. And there's bunches of them being named literally every day. Now you can say, well, okay, so there's these new Bartonellas, how important it is. We had these 2 isolates in our laboratory for 2 years, setting in a freezer because there were no sequences in GenBank that were comparable to the DNA sequences that we were getting out of these bacteria. And then Michael Kasoy, who's a researcher and a vector biologist at CDC, dumped sequences in GenBank that were from sheep kids, and David Bemis, who's a veterinary microbiologist that actually ordered sheep blood, poured his plates and had bacteria grow up on it and was smart enough to sequence those bacteria and then put those sequences in GenBank. So all of a sudden we've got KEDs on sheep, we've got sheep blood, and we've got 2 isolates in my lab that match 100%. And that was published in Emerging Infectious Diseases by Ricardo Maggi. Now where did our 2 isolates come from? They came from a patient in California that lived on a sheep farm with pericarditis, and they came from a woman in Ohio that got some unusual insect bite. She doesn't know what bit her. She got a rash-like lesion that was very large on her thigh, and then returned to North Carolina where she became involved in our studies. So this, I think, is important because there's a lot of Bartonellas out there. There's probably a lot of ways in which humans can become in contact with these organisms. In most instances, I believe epidemiologically, we clear these infections, and that's the end of the story. But in a subset of individuals, that is not the case based on our research that we'll get to here in a minute. So fleas, cats, and Bartonella species. Veterinarians are taught, unfortunately, that fleas are these obnoxious little things that make animals scratch. and induce flea allergy dermatitis in a subset of dogs and are the source of tapeworms. End of story. That is not the end of the story. And when I lecture to veterinarians, I basically tell them, kill fleas, kill fleas, kill fleas, because you will prevent a tremendous amount of disease in the cat, in the dog, and in the family, okay? So we've got to re-educate veterinarians, and we've clearly got to start educating physicians that part of the medical history is not only has there been exposure to fleas, but has there been exposure, to ticks, but has there been exposure to fleas? And again, looking at the data from Sweden, Norway, and Denmark, there is a big difference in the prevalence of Bartonella bacteremia in cats. So you can use that as an indicator where it's almost 40% in Denmark, and the studies from Norway and Sweden would suggest that it's a very low prevalence, assuming that you don't have too many fleas here too often, although some people have told me that there clearly are fleas here on cats. So the problem, again, for cats and fleas is that we know that they're transmitting henselae, cholerae, and chlamydia, and there's good evidence that they are probably transmitting Bartonella quintana as well. The French, Didier Rioul's lab has amplified quintana out of fleas, and we've isolated quintana from a human and from cats in North Carolina. So cats can get quintana and, So why does this become important? Again, my life is, over the last several years, has revolved around emails and phone calls from mothers of sick children many times. And this was another example in which this 18-year-old young girl had peripheral visual loss, sensory neuropathy, and a history of hallucinations, which I was really reluctant to put into the manuscript, but since it was part of the history, it was included. Initially, these were very infrequent and very non-bothersome. Over time, they became much more frequent, much more severe and bothersome to this young woman. Obviously, in addition to other physicians, she had seen a neurologist, a psychiatrist, and a neuro-ophthalmologist. And what you'll see on the other slide, and trying to help understand this was done retrospectively. So when we first started testing this young lady, we were doing just like everybody else. We were looking for Bartonella quintana, Bartonella henselae, and Bartonella vinsonii berkhoffii. So if you tested this woman against henselae, vinsonii berkhoffii, and quintana, she had no detectable antibodies. If you then developed an IFA test using a cat isolate that we had in the laboratory of Bartonella quintana, she had antibodies specifically against the Bartonella that we ultimately determined after developing a specific set of PCR primers for Bartonella cholerae that she was infected with cholerae. This initial result, we got a positive PCR, but we could never sequence it. And the policy for our research is unless we can get a DNA sequence that matches that band on a gel, we report it as indeterminate. Because it was indeterminate, she was never treated, so we followed her up for well over 801 days, which is in the publication that's cited here. She was treated by an ID physician at Duke, Chris Woods, and had total resolution of everything that I showed you on the previous slide. Ticks. So I had a PhD student that I was co-mentoring with one of our parasitologists, Sarah Billiter, and she was such an enthusiastic student that I requested that she review the world literature on Bartonella transmission, which she was very happy to do in her spare time. And that resulted in this publication because really what I wanted her to do is to review what was starting to appear in the literature supporting the possibility of tick transmission. And I didn't realize that when we did this, we'd get into so much trouble from the classical entomologists that are very particular in regard to mentioning tick transmission of new organisms. So that got us into a bit of a problem, and ultimately I can tell you that this went from yes and maybe with work being done by French veterinarians and suggesting that Oxodes rhizensis could potentially transmit Bartonella henselae, published in EID. And then the same group, in collaboration with Michael Levin at CDC, was able to prove this in a rodent model with Oxodes rhizensis attachment. So we now know in the laboratory transmission can occur. What we don't know is how often in the northeastern United States with Oxodes scapularis, Oxodes pacificus, or here with Oxodes rhizensis, there's actually people becoming infected. What we do know, if I press the right button, is that there are people with a history of tick exposure in Holland and in Denmark that we have isolated Bartonella henselae San Antonio 2 genotype from. And this is published in this particular manuscript. I'm sorry. Let me back up here. I'm looking over to this screen on the right, which is the next slide that'll come up. So these are two manuscripts that I would really suggest that you review. The first is from Volkart Kempf in Frankfurt, and in this study, they tested Oxodes rhenensis ticks from Germany, Spain, Portugal, and France, and essentially the prevalence of Bartonella henselae in those ticks ranged from 0 to 40%. That was done by 16S gene in his laboratory, and then done in my laboratory using the same extracted tick DNA, targeting a totally different gene, and the match between the two laboratories was about 95%. So sometimes we got positive and they didn't otherwise. So it's clear, and this was one of the earlier publications, but there's many subsequent publications, mostly out of Europe, identifying Bartonella henselae. Dr. Perrone mentioned this particular manuscript yesterday, which again is a documentation of Bartonella henselae by high throughput sequencing in ticks from France. And so I think there's good evidence that ticks have Bartonella henselae in them. What we need is good evidence that that's being transmitted to humans or animals. And this is, again, a situation where a human anesthesiologist got on the plane and flew her entire family to Raleigh, North Carolina to be tested in our laboratory. She had been sick, her son had been sick, and in the interest of time, I'm not going to go through their clinical histories. Both of them that were sick, we managed to isolate Bartonella henselae, San Antonio 2 genotype, and sequence that. But what was really important, and let me just back up for a second, is when I first read the literature from physicians that were writing about Bartonella, and they talked about these stria-like lesions being pathognomonic for Bartonellosis, I'll be very honest, I didn't believe it. I talked with Joe about this a little bit yesterday about how that originally appeared in the American literature. But this young man, and this is actually on his hip, he also had these on his waist, had stria-like lesions. He also had a blue nevus and actually bacillary angiomatosis. She got a surgical appointment, had the nevus removed, and biopsy disc. We PCR'd the nevus. It was negative. We couldn't find Bartonella henselae DNA. This is the histopath from the stria lesion, where you can see around the blood vessels there's a lymphocytic plasmacytic infiltrate. These tissues were then sent to Marna Erickson, who's in the Department of Dermatology at the University of Minnesota Medical School. Marna's worked very hard on using confocal immunohistochemistry, and it'll be hard to see with the lights, but what this is a patient with bacillary angiomatosis, so this is the positive control. This is the negative control, in which we did not see any immunofluorescent organisms. What this depicts are little individual Bartonella organisms at the end of each arrow. What's also important here, and really needs to be studied, but as the discussion with biofilms yesterday, if you use a gram-negative stain, there are other bacteria that light up, presumably in this biofilm, that will not light up with a Bartonella-specific probe. And so what this means on the basis of an N of 1 is anyone's guess. If you stain the fluorochrome and you put a different one on, again, here they're lighting up green rather than red, but I think it's some convincing evidence that we can find these bacteria within these lesions. I've suggested in the United States, if there's one study that really needs to be done, it's a study to definitively determine whether this is a pathognomonic lesion associated with Bartonellosis, as we associate ECM with Lyme disease. Whether that'll occur is yet to be determined. So for those of us who are clinicians, every once in a while you have one patient that you either never forget, or in this case, it completely changes the direction of your research. And so this dog, at the point this picture was taken, was 3 years old, named Tumbleweed, and over a 1-year period, from about 2 years of age to the time it was referred to the university, this dog had a 6-month history of shifting leg lameness and polyarthritis. Over the next month, it lost 10 pounds or 5 kilograms, developed seizures, vasculitis, that was proven by a biopsy, which looked like a leukocytoclastic vasculitis that's been reported in children with Bartonella henselae infection in the human literature. Then went on to develop epistaxis. So we've done subsequent studies in dogs published in the Journal of Clinical Microbiology, and I say this on purpose because if I were a physician and I had a young child with epistaxis of undetermined etiology, this genus of bacteria would now be on my differential list. And then finally, due to immunosuppression, so anti-nuclear antibodies came up yesterday. We've documented anti-nuclear antibodies are prevalent in dogs with Bartonella infections and Ehrlichia infections, as are anti-neutrophil cytoplasmic antibodies. This veterinarian consecutively immunosuppressed this dog, and it's the reason we were able to make an isolate. I am fairly well convinced with the techniques we were using then, because I then sacrificed 6 years in a graduate student trying to make other isolates, and after 6 years of effort using clinical patients coming through our hospital, she had one more isolate from that case of an hemangioparasitoma. So we then figured out isolating Bartonella from a dog is very difficult. Isolating Bartonella from a cat is very easy because cats maintain a level that's somewhere between 1,000 and 10,000 times higher than a dog, horse, or a human, okay? And I can say horse because we've done that work. So we ended up with this isolate from this immunosuppressed dog that then went on to develop endocarditis. If you look at the reviews we've written, Duke and the Mayo Clinic have both repeated this experiment by taking human patients with anti-neutrophil cytoplasmic antibodies, immunosuppressing them, and having them come back to their respective institutions with endocarditis. And again, that wasn't work we were involved with. These are case reports in the human literature. But it gave us a new isolate to make an antibody to. And then we started emphasizing, what is this new bacteria, Bartonella vinsonii berkhoffii, doing? We came to the conclusion pretty quickly that trying to find Bartonella in a dog was going to be a challenge. We could find a very low seroprevalence to this organism in our clinical patient population going through the hospital, but we were unable to make isolates using the same approach that we had used, which was a lysis centrifugation approach, successful in cats. So Dr. Sushama Santaki was a postdoc in the lab, and this is the way we spent our Christmas in 2003. And the idea we came up with in a lab meeting is maybe if you use an insect biochemical composition media rather than a mammalian growth media, you might make these things grow better. And the reason we came up with that idea is researchers around the world were saying fleas, lice, ticks maybe, biting flies, and others. So that was the reason for the idea. And the good news is the answer to the question was yes, that will actually increase your ability to detect these bacteria. Ashley Duncan, the next PhD student that entered into the lab, used this approach, and initially we did the validation through testing sick dogs coming through our teaching hospital, and we referred to this as the BAPGM platform. And it, as I'll show you in the subsequent slides, which are all studies related to humans, what we do is every sample, we extract DNA, we target an intergenic spacer region. Again, this is the same region we used on the ticks that Kemp sent us from Germany. We put this into tissue culture at 7 days, 14 days, and in horses 21 days, because in a horse we will not find this until it's incubated for 21 days. Problematic, certainly. And then we subculture, and for all Bartonella researchers, there are very few isolates in the entire world. So getting this thing to come out of a liquid culture and grow on a plate is still a challenge that we have not been able to overcome. And what you can see then is anything that I tell you about from here out is documented on the basis of DNA sequencing in a commercial lab with us blasting those sequences and comparing it to our Bartonella database. So this is where the conflict of interest occurred in my life, because NC State looked at this idea and they said, gee, this is a really neat, novel idea. Nobody's ever done this. The university said we should patent this, and so they filed for the patent. I tried to get several companies, I know people both on the human diagnostic sides and the veterinary diagnostic side, to fund research or to embrace this or take the technology and do something with it. And the answer was no, no, and no. And ultimately, for reasons I won't bore you with, we ended up starting a company during a recession to try to diagnose a single genus of bacteria that most physicians had never heard of and most veterinarians were just starting to learn about. So why is this important and why did we go forward with this effort? One of the reasons is because this veterinarian worked at our institution. This veterinarian had gotten sick, and it was pretty obvious as you walked down the hallway that something was going on because the faculty rumor mill was, does this person have cancer or does this person have AIDS? What's going on? Have you noticed this type thing? I had no clue until the person came and talked to me and said, Ed, I've been going to UNC, to the medical school, to the neurology department for about 9 months. And in the last 3 weeks, I've had to pull my daughter out of school because she too has become sick, and our lives and family, things are kind of falling apart. The good news is my wife and the other 2 sons are still healthy. So we tested the wife and 2 sons, they were negative, and we tested the father and the daughter that I'll show you on the next slide. The good news, and again, one of the problems with detection of Bartonella is we always PCR from blood, we always PCR from serum, and we always PCR from enrichment culture because we will have negative enrichment culture as seen here. Now you see what happens is once we got this, he went back to UNC to the medical school, and the neurologists were kind enough to supply blood and cerebrospinal fluid. And the bad news, and we've only tested a few human cerebrospinal fluids, but what I can tell you is that there is no increase in protein and there is no increase in cells when we can grow Bartonella out of cerebrospinal fluid. Obviously, this is a very small number of patients. We need a whole lot more data, and whether that's uniformly true or only partly true, and I think it'll only be partly true, but I can tell you that his cerebrospinal fluid report, which is in our files, was absolutely normal. You can see that we got growth in enrichment culture from both. And what's also interesting, so in the veterinary literature in cats, there's some evidence cats get this very severe periodontal disease that ultimately is so refractory to treatment that veterinarians just pull all the teeth out. It sounds barbaric, but sometimes the cat's a whole lot better off and a whole lot healthier. This veterinarian said, I've had periodontal disease, and we ended up testing swabs and ended up showing that those swabs from a periodontal swab, we could amplify and sequence Bartonella vinsonii berkhoffii. So I think this slide's really important. It's certainly important to me as a veterinary researcher in the context. I don't think CDC targets over the years would have ever proven by direct PCR only through enrichment culture that we were consistently able to demonstrate that she was infected with the same Bartonella vinsonii berkhoffii genotype that came out of her father, which is not the genotype that came out of tumbleweed. There's 4 different genotypes. You all have genotype 3 here in Europe. You actually have it in German Shepherds that you ship to the United States that go to Texas and develop endocarditis and then are tested in my laboratory, purchased at very high prices by the U.S. military. We have genotypes 1 and 2 in the United States more often than we have genotype 3. Okay, so after that experience, I started giving lectures on what was exciting to me, Bartonella in dogs. So granulomatous hepatitis in a dog, granulomatous lymphadenitis, and different other cases that came into the hospital. And at the end of those lectures, if there were 100 veterinarians in the audience, I would have 3 to 6 come up and say, I've been sick for 3 years, 6 years, 10 years, and I've been to a lot of doctors, and I don't really have a diagnosis. Or I have a diagnosis of MS-like disease, rheumatoid-like disease, something of this nature. So we started testing veterinarians. So that's how a veterinary researcher went from cats, dogs, horses, and some marine mammals to looking at sick veterinary workers. So this was the first 6 that we published in the Journal of Clinical Microbiology, and I'm just going to emphasize this one young lady up here with seizures. She had a diagnosis of cat scratch disease by a physician 18 months before we tested her, and a year before she started developing seizures. Her initial seizures were infrequent, but had progressed to grand mal seizures and cluster seizures and were quite frequent. You can see that we didn't get Bartonella vinsonii berkhoffii, and we didn't get Bartonella henselae San Antonio 2 out of her, We got the Houston 1 strain, which is a strain that came out of an AIDS patient in Houston, Texas, and is the type strain for Bartonella henselae. You can see here that we got this by PCR. Again, our BAPGM culture failed us. These antibodies were done by William Nicholson at the CDC. At that point, we were doing serology in collaboration with the rickettsiology group at CDC, so all these antibody titers are CDC titers. Once we got this result, we brought her back, so she's positive in blood, in culture. And then what ends up happening, and we also got an isolate, which was very nice because we don't get them very often, is 256 days later, her mother calls me, and they refused to treat her on the basis of data that came out of a veterinary research lab. So 256 days later, she was referred back to Chris Woods at Duke, where they obtained cerebrospinal fluid. I am an adjunct professor at Duke, and some days I have a love-hate relationship with the organization because they don't always get it right. And they were supposed to send blood, they were supposed to get serum, but we did get CSF that day. I'm not sure where the other samples ended up, but what was fortunate is we were able to demonstrate exactly what we had found on day 31 and day 0 when we studied this girl. That with antibiotic therapy, she was cleared. She, like the Herxheimer reaction, one-and-a-half-year period after antibiotic therapy. And I'll show you one other case where we've been involved more recently. I think yesterday, Richard showed this particular slide, and this, in my opinion, is the most disconcerting thing that a email or phone call has resulted in. And in this instance, published in the Journal of Clinical Microbiology, we had a father, mother, and son, and a daughter that died at 8 days of age. So we were able to work with the human pathologist that did the autopsy in New York on this particular patient and demonstrate in her tissues at 8 days of age, multiple tissues, multiple PCRs, multiple sequences, and anytime we do this, we separate those tissues, so we do one a week for about a 4 to 6 week period. So if somebody's going to criticize DNA carryover, they're going to have a lot more trouble criticizing it on the basis of the fact we didn't process all the tissues at one time. We isolated the same 2 Bartonellas 10 years later out of the mother and the son. And the father, and this has been alluded to in regard to Borrelia burgdorferi transmission, but whether there's direct transmission or sexual transmission of Bartonellas is clearly yet to be determined. There is some evidence of spouses of individuals that have had classical cat scratch disease ultimately being infected with these bacteria, but the evidence is minimal at this point in time. And you can read the family histories in the J. Clem Micro manuscript if you're interested. Now, in veterinary medicine, we haven't had to be too concerned about needle stick transmission because we don't deal with hepatitis viruses, and we don't, at least we don't think we do. The Dutch may change that story pretty soon. And we don't deal with HIV. We certainly deal with some other retroviruses, but there's no evidence that they've ever been transmitted to a human. With Bartonella, this is actually a veterinarian institution that was tested by us because the dog that bit this woman, or the dog that she actually stuck the needle into the tumor and then stuck it into her hand, was a very fractious dog. And the question from the hospital administrator was, could she have gotten Ehrlichia from the dog? What I knew and he didn't know is that 36% of the dogs in the southeastern United States that have antibodies to Ehrlichia have antibodies to Bartonella, vinsonii, berkhoffii, versus 1% of healthy dogs that we've tested and 3.6% of sick dogs going through the veterinary teaching hospital, about 3,000 dogs that we tested. We, that PhD graduate student that spent 6 years looking for another isolate, also tested those 3,000 some dogs. So the prevalence is really low until you find dogs that have gotten tick transmission of Ehrlichia. So my concern was, might she have Bartonella from the outset? Is we've got the first culture from her when she was absolutely healthy at 5 days, no problem, everything was negative. At day 33, again, we were able to get it out of a serum sample, not out of her blood, not out of enrichment culture. She still had no antibodies. At that point, we retested her a couple times and she was negative. At day 81, we retested her again and now she's culture positive. and she still only has an antibody titer of 1 to 32. So if it takes Borrelia burgdorferi at least 6 weeks in most people to develop an antibody response, I'm not sure how long it actually takes in a human to develop an antibody response, but it was somewhere between 81 and 234 days that she finally seroconverted to a titer of 128. Typically, Bartonella has very, very low antibody titers. Now, I'm going to review 2 of the major case studies that we've been involved with. The first was high-risk veterinary patients. The first here that I'm presenting is the rheumatology patients. This was with a single rheumatologist, Robert Mosiani, in the Rockville, Maryland area. The next group is 192 patients that are the high-risk patients that are predominantly veterinary workers. And if you want to figure out whether Bartonella is a problem in Scandinavia, it's a very simple population that should be studied. Start with forestry workers and veterinary workers and healthy people, and then see what you find. And then finally, thanks to Chris Woods and others at Duke, we studied, 32 healthy medical students and physicians that volunteered blood from the medical school. And again, only one of those individuals had antibodies in our testing, and none of those individuals in the control group were BAPGM positive in any aspect of the testing. All were negative. So what did we find in summarizing, comparing, contrasting these 2 studies? And in the interest of time, I'm just going to drop down to these results. In blood donors in the United States, it's about 3.6%. In veterinarians, based on CDC testing, it's between 6 and 9%. In forestry workers, those studies have varied anywhere from 10 to about 40% in and around the world. In this group, we got almost 50%. Amazingly enough, with Dr. Mosiani selectively picking patients that he considers to have small vessel disease, an increase in C-reactive protein, or other acute phase reactants, he got a higher seroprevalence than if I tested sick veterinarians that came up to me at the end of a lecture. And that was most unexpected. He also was able to, in our testing, again, done by Riccardo Maggi in our lab, who's a microbiologist, molecular biologist, come up with bacteremia. So we do believe that this organism needs to be investigated in regard to neurologic disease, in regard to microvascular disease, and in regard to arthritic disease. Finally, similar to what's been said with Lyme disease, but much, much worse, in both dogs and people, at least half of the ones that are bacteremic, and what you'll see up here on this particular slide is if you just look at this box here, these are people that are bacteremic, but they're IFA negative. And as you look down, 82% of the ones infected with Bartonella henselae by BAPGM enrichment culture did not have detectable antibodies by our testing. And that's been consistent whether it was testing done by us, by CDC, or by the Mayo Clinic. So as we say in the United States times, Houston, we've got a problem. And then finally, I wanted to finish with this young lady because I was asked to be on a master's student in Grenada's work, which has been recently submitted that has nothing to do with Bartonella. I was there less than 24 hours when they talked to me about one of their veterinarians that had graduated a year and a half before, who had just a horrendous history that's detailed in this publication in Parasites and Vectors. She had seizures, grand mal seizures in JFK Airport, which I had a hard enough time getting through JFK, getting over here. So if I seizured there, I think it would be a problem. She was also medevaced off of Grenada to Trinidad and put into a phenobarbital-induced coma for 14 days before they could get her out of the coma. Yes, and I think you can relate to some of the experiences this young lady, when I met this girl, I couldn't tell how much was disease and how much was drugs because she was on 6 or 7 different drugs at that point in time, most of which I've never used as a veterinary clinician and most of which I had to look up to figure out what type of drugs they actually were. These are drugs you all are familiar with, but I was not. The bottom line is she was co-infected with 2 organisms that we know an awful lot about in veterinary medicine, a anaplasma species that infects platelets called cyclic thrombocytopenia in dogs, and a hemotropic mycoplasma. So I'm not talking about hemotropic mycoplasma today, but a lot of this work has been done in Germany, in England, and in labs in the United States. We've known about these organisms in veterinary medicine for many years. They are not highly pathogenic organisms, but they are cofactors. What I would tell you is that there are 32 PCRs done on this young lady on 8 enrichment cultures, and we only found Bartonella, henselae in 1 enrichment culture at 14 days, which is embarrassing and disconcerting, but it's how hard these bacteria can be to find. This girl was treated with 6 months of doxycycline, and at the end of the 6 months, she was still PCR positive in our laboratory. She was then treated with doxycycline rifampin, got married last December, and has not seizured for almost 1 year. So hopefully you will think about the next patient that you see that's having seizures and at least think about the possibility that some of these vector-borne organisms are able to persist for long periods of time and perhaps cause very adverse findings in both our veterinary patients as well as human patients. So what have we learned about Bartonella? Seronegative infection is common. This manuscript by the current PhD student published in JClim Micro. When we do 3 cultures now, so from neurologic veterinarians, I started doing Monday, Wednesday, and Friday, or at least 3 a week. We never had any veterinarian amongst this group that was positive in all 3 cultures, and we only had a couple that was positive in 2 out of the 3. Most people were only positive in 1, supporting the fact that people are probably having a relapsing bacteremia or that detection is so low. Treatment failures are common, and they're common with 3 months of doxycycline rifampin, and my current suggestion is we don't know how to treat this infection in a cat, dog, horse, or human. Unlike Borrelia burgdorferi, Bartonella has bacteriophages, probably somewhere between 400 to 600 viral particles per bacteria. What role this might play in disease or chronic disease is yet to be determined, but I think it's very interesting that we've got a bacteria that can live within our endothelial cells, circulate through our body in erythrocytes and macrophages, and are carrying a whole bunch of bacterial viruses around. And then Richard's alluded to the importance of co-infections. We've also agreed. So the message I wanted to relate, and Dr. Perrone brought this up yesterday at the conclusion of his talk, is we really need a One Health and team approach to studying and understanding vector-borne diseases. We need physicians, veterinarians, entomologists, and everybody working together, not in silos, because by working together, I think we'll get to better answers much more quickly for our patients. And with that, I would suggest that for the veterinary profession, Bartonella is the most important One Health genus of bacteria that we have to sort out over the next several years on the basis that we've got large animal reservoir, we've got numerous vectors transmitting, and we're having a jump across species that's inducing very complex and very presumably chronic diseases. I want to acknowledge the people in my laboratory that actually do the work. Right? They have been a great group, and right now I have the best group I've ever had in the lab, and I thank you for your attention, and I think we'll probably end up doing questions later. Thank you, Carol.